Photosynthesis in plants, non-cyclic photo-phosphorylation. Plants are the most fascinating organisms on the earth. They are autotrophs, which means they cook their own food by photosynthesis, in the presence of sunlight and chlorophyll. Okay, but where do they cook their food? Basically in leaves. We simply call the leaves as food factories of plants. Leaves consists of mesophyll cells which look like a typical plant cell consisting of chloroplasts. And these chloroplasts, also called plastids, are the actual site of photosynthesis. A chloroplast is a double-membraned organelle consisting of stacks of thylakoids. A bunch of thylakoids stack one above the other forming granum. All such granum are together called a grana. If we zoom into a thylakoid, this is how it looks like. A membrane sac-like structure with a lumen at the center. Let's now go for transverse cross-section of the thylakoid to understand it better. Here comes the thylakoid consisting of a membrane shown in a color of light green gradient, whereas the lumen is shown here in dark green color. Basically, a thylakoid consists of various pigment systems, protein complexes, few electron carriers and acceptors distributed across the membrane and the lumen. The light-harvesting pigment system, and photosystems of both, the PS1 and PS2 along with the plastoquinone, PQ, and cytochrome chamber are present on the thylakoid membrane, while the photolysis of water-occurring oxygen-evolving complex, OEC, is located inside the lumen just next to the light-harvesting complex 2. Every photosystem consists of two components. One is a pigment system with a reaction center called LHC, the light harvesting complex, and the other one is a complex of proteins and electron acceptors called the photosystem. The first discovered photosystem is called PS1, and the later discovered is called PS2. In the same way, their respective light harvesting complexes are called LHC1 and LHC2 respectively. The non-cyclic photophosphorylation begins with the LHC2, ejecting a pair of electrons from its reaction center, also called P680, by absorbing a sunlight of wavelength 680 nanometers. The energy present in the quanta of photons of sunlight is utilized for the excitation of electrons, and so, they eject from LHC2. And are accepted by a primary electron acceptor called phosphodin protein complex, which is a part of PS2. From this PS2, the electrons are further carried to the PQ, plastoquinone. This PQ acts as a gate for the electron's entry into the cytochrome chamber, as well for the entry of H+ proton molecules into the lumen. Once the electrons reach the PQ, plastoquinone, utilizing their energy, the PQ pulls the H+ from stroma of the chloroplasts into the lumen of the thylakoids. This process continues, with every pair of electrons pulling a pair of H+ into the lumen. Due to this continuous influx of H+, a lot of H+ are concentrated inside the lumen. This creates a concentration gradient of H+ across the thylakoid membrane between the lumen and the stroma. Meanwhile, the pair of electrons proceed further from PQ to cytochrome chamber, and via the cytochrome, the electrons will be carried forward to another electron carrier, PC, plastocyanin, which is a copper chamber. Thus the electrons are now at plastocyanin, which are again carried forward to LHC1, the reaction center of photosystem 1. Owing to the uphill and downhill transport of these electrons from LHC2 to LHC1 via the PS2, PQ, cytochrome and PC, the electrons are now in a de-excited state and need sunlight energy again to move further. Here comes again, one more quanta of photons of energy with a wavelength of 700 nanometers. This energy is absorbed by the reaction center, P700 of LHC1, and again the pair of electrons are ejected all the way to PS1. PS1 is a complex of proteins where, finally, the electrons are accepted by an enzyme ferridoxin, which is present inside a complex called FRS, the ferridoxin-reducing substance. This complex is made up of iron, Fe and sulfur S, and so sometimes also called an iron-sulfur protein complex. However, the purpose of these electrons is to synthesize NADPH2, an assimilatory power of photosynthesis besides ATP. Hence these 2 electrons will be further carried to another complex, called NADP reductase chamber, where the 2 electrons along with a pair of H+ protons fuse with NADP+ to reduce it to NADPH2. Now we should get back to lumen, where the concentration gradient of protons H+ is established. As a lot of H+ are densely packed in the lumen, they generate a very high energy potential called PMF. Now all these H+ with this PMF try to move out of the lumen to stroma so that they can balance the concentration of protons on either side of the thylakoid. But there is only one pathway out, and that is the ATPase or ATP synthetase enzyme, also called F0 and F1 particle. Now all the H+ try to move out through this F0 and F1 particle enzyme, during which the energy potential of H+ will be utilized by the ATPase particle to fuse ADP with an inorganic phosphate and form ATP. Thus phosphorylation, which means ATP synthesis, happens. Now, the light harvesting complex of PS2 have to be compensated with the loss of electrons, so that the continuous supply of electrons is assured for NADPH2 synthesis. This can be achieved by photolysis of water inside the oxygen evolving complex. Oxygen evolving complex, OEC, is located inside the lumen, just beside the LHC2. Here one molecule of H2O splits into 2H+, 2E- and half O2. Now this 2E-, which are a pair of electrons, compensate the LHC2 of PS2. Hope you all understood non-cyclic photophosphorylation. Thanks for watching. Please subscribe our channel.